Glucose and its derivatives have different functions in living organisms. One of them is generation of energy during its breakdown. Some of the energy can be saved in the form of other molecules. The process of glucose breakdown to pyruvate, which is accompanied by the synthesis of ATP, is called glycolysis or the Embden-Mergov-Parnas pathway. Mostly it takes place in the cytoplasm of cells. This process is traditionally divided in two phases. The first phase is the preparatory phase. During the phase, glucose is converted into less stable intermediates, which in the final phase results in the formation of phosphorylated trioses, glyceraldehyde 3-phosphate and dihydroxyacetone monophosphate. Its consideration is commonly started from the reaction of glucose phosphorylation, which is catalyzed by the enzymes that have the general name of hexakinases. The donor of the phosphoryl group is ATP. The product are glucose 6-phosphate and ADP. Glucose 6-phosphate is an intermediate of some other metabolic pathways, such as gluconeogenesis, pentosophosphate shunt, glycogenesis, glycogenolysis, the synthesis of glycosaminoglycans. Hexakinases are the regulatory enzymes for many of these processes. Phosphorylation of monosaccharides in cells anchors them in the cells and directs the sugars to metabolic processes taking place in the cells. The next reaction is catalyzed by phosphohexose isomerase. Glucose 6-phosphate is converted to fructose 6-phosphate. Fructose is phosphorylated to form fructose 1,6-bisphosphate. The enzyme that catalyzes this reaction is phosphofructokinase. In most cases, the donor of the phosphoryl groups is alpha ATP, and such phosphofructokinase is called phosphofructokinase type 1. It is one of the key regulatory enzymes of glycolysis. In some plants, pyrophosphate can be the donor of the phosphoryl groups. Fructose 1,6-bisphosphate decomposes to glyceraldehyde 3-phosphate and dihydroxyacetone monophosphate. The reaction is catalyzed by aldolase. These three carbon molecules can be interconverted, and this conversion is catalyzed by triosophosphate isomerase. This is the end of the first phase. In general, glyceraldehyde 3-phosphate formed directly from fructose 1,6-bisphosphate or via dihydroxyacetone monophosphate will be the focus of the next phase. Summing up the first phase, we can say that the gradual phosphorylation of the 6 carbon molecules reduces its stability and leads to its breakdown with the formation of 2 trioses. Although glycolysis, as mentioned earlier, generally produces ATP, this phase takes 2 ATPs for each glucose molecule. If you want to get gain, you need to invest. So we got 2 glyceraldehydes in the previous phase. The second phase is called the payoff phase. This is the phase that produces ATP and NADH. The first reaction of this phase is the oxidation of glyceraldehyde to 1,3-bisphosphoglycerate, which is accompanied by the consumption of inorganic phosphate. This reaction is also accompanied by the transfer of electrons or oxidation from glyceraldehyde 3-phosphate to NAD+, with the formation of NADH. We had 2 molecules of glyceraldehyde 3-phosphates, so we got 2 NADHs. This reaction is catalyzed by the enzyme glyceraldehyde 3-phosphate dehydrogenase. 1,3-bisphosphoglycerate can donate its phosphoryl groups to ADP with the formation of ATP. This reaction is catalyzed by phosphoglycerate kinase. We had 2 molecules of glyceraldehyde 3-phosphate, so we got 2 molecules of 1,3-bisphosphoglycerate, each of which donated its phosphoryl groups to ADP. Therefore, we have obtained two ATPs, which makes up for the two ATP molecules that were spent during the first preparatory phase of glycolysis. The product of this reaction, in addition to ATP, is 3-phosphoglycerate. The way of ATP biosynthesis by transferring a phosphoryl group from another compound such as 1,3-bisphosphoglycerate to ADP is called substrate level phosphorylation. 3-phosphoglycerate is converted to 2-phosphoglycerate. This is catalyzed by the enzyme phosphoglycerate mutase. 2-phosphoglycerate is converted and dehydrated by enolase to phosphoenol pyruvate. Phosphoenol pyruvate is another compound capable of donating its phosphoryl group to ADP with the formation of ATP. From 2-glyceraldehyde 3-phosphates, we gradually got 2-phosphoenol pyruvates, and thus the next two molecules of ATP. The reaction is brought about by pyruvate kinase. It is also a regulatory enzyme of glycolysis. This is the last reaction of glycolysis. The product of this reaction is pyruvate. During this phase, we obtained 4 ATPs, 2 NADH, and 2 pyruvates. In total, however, taking into account those 2 ATPs that were spent during the preparatory phase, glycolysis gives 2 ATPs, 2 NADH, and 2 pyruvates for each glucose molecule. During glycolysis, we have oxidation and glucose breakdown. From the point of view of thermodynamics, under standard conditions, the oxidation and splitting of glucose to 2 molecules of pyruvate is accompanied by the release of energy, 146 kilojoule per mole, which is sufficient and can be potentially used for the process that requires energy, namely the synthesis of 2 ATPs from ADP and inorganic phosphate. The fate of pyruvate may vary depending on the condition occurring in cells. For example, it can be converted into so-called acetylcoenzyme A, which then can be oxidized in the Krebs cycle and yield even more ATP by oxidative phosphorylation under aerobic conditions. By the way, under this condition, NADH, which was formed during the second phase of glycolysis, can also contribute to the synthesis of ATP. Under the limited oxygen supply, pyruvate is converted to lactate, simultaneously liberating NADH, NAD plus from NADH, which is then reutilized in glycolysis. This is sometimes called anaerobic glycolysis. Pyruvate can be used to synthesize something else, such as oxaloacetate, which is necessary to support the Krebs cycle. But all the above mentioned processes do not belong to glycolysis.